glycogen. But before that, make sure you know how a glycogen structure looks like. Glycogenolysis requires two basic steps. The first one is the removal of glucose from the non-reducing ends of glycogen on the outer branches by glycogen phosphorylase. Next is the hydrolysis at branch points of glycogen by glycogen debranching enzyme. Kalau kita tengok di sini, yang merah ni itu adalah non-reducing end of the glycogen. Yang hijau tu adalah the branch point of glycogen, and the gray beads, those are glucose. Now let's look at the glycogen phosphorylases. Glycogen degradation is initiated by the action of phosphorylase. It's an enzyme that which it will break the alpha-1,4 bonds, and then it will insert a phosphate at carbon 1 to the non-reducing end. Apa kita dapat? Glucose 1-phosphate. That action will be repeated on and on and on and on and on and on sampailah it will stop at 4 glucose residues before that alpha-1,6 junction. As a result, you can see we have 1, 2, 3, 4 glucose. Then what? Okay, another enzyme, oligo-alpha-1,4 glucan transferase, will separate trisaccharide from the terminal branch. That trisaccharide, also known as limit dextrin, will be transferred to the end of a neighbor branch. Di sini, it will attach via a alpha-1,4 bond. Maka tinggallah a single glucose with an alpha-1,6 bond. Then what? Now we have the second one is hydrolysis of alpha-1,6 glycosidic bond. This is catalyzed by alpha-1,6 glucosidase or debranching enzyme, which releases free glucose. In conclusion, the concerted action of phosphorylase, gluco-alpha-1,4 glucan transferase, and also alpha-1,6 glucosidase releases glucose 1P and some free glucose molecules. Okay, remember that the branching enzyme causes hydrolysis dan bukan phosphorylasis. Also note that only glucose in the branching position is released as free glucose. Yang lain-lain semua dalam bentuk G1P. On average, a free glucose molecule is produced for every 9 glucose of glucose 1P, which that can help us to identify the degree of branching in a glycogen molecule. Now what happened to G1P? It will be converted to G6P by phosphoglucomutase. This is the reverse reaction in glycogenolysis. Finally, the last stage of glycogenolysis is the hydrolysis of G6P to glucose and inorganic phosphate. This will be catalyzed by G6-phosphatase. The enzyme is found in liver and kidney cell membranes and also in intestinal cells endoplasmic reticulum, but not in muscle cells. That is, muscle cell cannot release glucose to the circulation, tetapi for liver, kidney and intestine, they can. Glycogenolysis in the liver is controlled partly by hormones. One of the important hormones regulating glycogenolysis in the liver is epinephrine or adrenaline. Epinephrine does not enter the liver cell. It actually binds to a receptor on the hepatocyte surface, and there is a second messenger is produced within the cell. The receptor for epinephrine is a G protein coupled receptor ataupun GPCR. That receptor is coupled to a protein. Let's call it G protein. This G protein will stimulate adenylyl cyclase, di mana this enzyme will increase the cyclic AMP. Cyclic AMP inilah the second messenger within the hepatocyte. Apa cyclic AMP buat? Dia akan activate an enzyme, protein kinase A. Pernah dengar kan? Protein kinase A. This protein kinase A, as we know, will start the phosphorylation and will cause glycogen synthesis to shut down, thus activating the glycogen breakdown. Okay, bagaimana nak return to inactivated state? Ada dua cara. Ada satu enzim, cyclic AMP phosphodiesterase. So enzim ni will degrade cyclic AMP tadi tu to form back into AMP, adenosine monophosphate. This will turn off the second messenger signal. That is nombor satu. Yang kedua, kita ada protein phosphatases. Protein phosphatases tu, fungsi dia ialah to dephosphorylate. Semua protein yang were phosphorylated earlier on can be dephosphorylated balik. So itu caranya untuk change the system into inactivated state. Kesimpulannya, actions of glucagon on target tissues, glucagon promotes glycogenolysis in liver cell, satu. Yang kedua, glucagon stimulates lipolysis during fasting. Macam mana dia buat? They activate the hormone sensitive lipase in adipose tissue. These will release the NEFA ataupun non-esterified fatty acids into the blood for further beta oxidation. Ini dah masuk topik lipid lah. That beta oxidation will be followed by ketogenesis and gluconeogenesis in the liver. Satu lagi, glucagon also increases the level of phosphoenolpyruvate carboxykinase, yang enzim yang terdapat di gluconeogenesis in liver cells. The carbohydrate metabolism does not work alone or stand alone. It functions together with other pathways and is orchestrated in such way to ensure the homeostasis in our body. Question: who created